D-ribose is the biologically active form of the naturally occurring sugar ribose, and it's produced in the body from glucose. Ribose is involved in the synthesis of ATP, which provides energy to the muscle cells during exercise. Supplementation with ribose has accelerated ATP synthesis following its depletion during intense exercise. A control trial in 12 male recreational bodybuilders found supplementing with 10 grams of ribose per day for 4 weeks resulted in greater gains in muscle strength and endurance than a placebo. D-ribose can also help to combat fatigue, and it improved mood and vitality in aging adults, and this can allow for increased exercise frequency. A dosing study found taking D-ribose on an empty stomach leads to more efficient absorption than by taking it with food. Periodically, concerns arise regarding the potential of D-ribose to promote damaging glycation reactions. While ribose can contribute to glycation reactions when present in high concentrations, the amount of D-ribose attained through supplementation is not worrisome. A growing body of evidence supports the use of omega-3 fats to improve recovery from strenuous exercise. Omega-3 fatty acids, particularly EPA, can be beneficial in the prevention and the treatment of sarcopenia. In a controlled study in older adults, daily supplementation with omega-3 fatty acids containing over 1.8 grams of EPA and 1.5 grams of DHA increased the rate of muscular protein synthesis compared with a corn oil. CoQ10 is an essential component of a series of biochemical reactions that generate energy in the cell's mitochondria. CoQ10 also functions as a free radical scavenger, protecting cells against oxidative damage. Clinical studies have demonstrated an exercise-enhancing effect of coenzyme Q10 supplementation. In a study in trained and untrained individuals, supplementing with 100 milligrams of CoQ10 for 2 weeks increased the length of the time of the participants could exercise before reaching exhaustion. A randomized control study in male runners found 14 days of CoQ10 supplementation reduced the spike in blood levels of lactate, interleukin-6, tumor necrosis factor alpha, and C-reactive protein induced by a bout of middle-distance competitive running. The dose of CoQ10 used in the study was 5 milligrams per kg per day, or about 350 milligrams per day for a 155-pound person. In an animal study, rats were supplemented with CoQ10 for 6 weeks during exercise training. This produced beneficial changes in levels of key regulatory proteins, including nuclear factor kappa B and NRF2, both of which defend against inflammation and oxidative stress. Arginine is a conditionally essential amino acid, and it participates in a variety of metabolic pathways, including protein synthesis. Importantly, arginine is a precursor of nitric oxide, a potent vasodilator. Arginine supplementation can increase blood flow to the muscles. In a controlled clinical trial in competitive male cyclists, supplementation with 6 grams of arginine daily for 3 days increased 20-kilometer time trial performance, reduced oxygen consumption, and reduced systolic and diastolic blood pressure. In another controlled clinical trial in untrained college-aged men, supplementation with a product containing 1.5 grams or 3 grams of arginine along with grapeseed extract for 4 weeks reduced the time to the onset of cycling-induced fatigue when compared to the placebo. Animal studies indicate arginine supplementation might be beneficial for exercise recovery. In one study, L-arginine supplementation before a single bout of exercise reduced muscle fiber damage, and it maintained exercise performance capacity in rats. These effects were attributed to increased muscle nitric oxide content. Resveratrol is a polyphenolic compound found in plants and plant foods such as grapes, red wine, peanuts, and Japanese knotweed. Resveratrol has been shown to favorably influence several factors involved in chronic degenerative diseases like inflammation, insulin sensitivity, oxidative stress, and endothelial dysfunction. There is clinical and preclinical evidence that resveratrol can augment the effects of exercise on muscle mitochondrial capacity, increasing energy production and utilization. In a double-blind, placebo-controlled trial in healthy young adults, daily supplementation with 500 mg of resveratrol plus 10 mg of piperine, a black pepper extract, combined with low-intensity endurance exercise for 4 weeks, significantly increased muscle mitochondrial capacity. 2 animal studies found resveratrol supplementation improved exercise performance compared with exercise alone. In one study, rats who were fed a diet supplemented with resveratrol during 12 weeks of exercise training were able to run longer and further than rats who were trained without resveratrol. Improved muscle strength was also noted in the resveratrol-treated rats. Gynostemma pentaphyllum, or xiaozhulong, is a herb with a long history of use in Chinese medicine as a health tonic. It's known as the herb of longevity. Components of gynostemma have been shown in pre-clinical research to activate AMPK, a major regulator of glucose, fat, and energy metabolism in the body. Animal studies have demonstrated the anti-fatigue effects of xiaozhulong. In one of these studies, the polysaccharides derived from the gynostemma extended the exhaustive swimming time of the rats. The gynostemma polysaccharide extracts also lowered blood lactic acid levels and increased liver and muscle glycogen concentrations. A study in mice found that the prolonged time to exhaustion from exercise after the administration of the gynostemma polysaccharides was linked to reduced oxidative stress and enhanced muscle glycogen levels. Cordyceps sinensis is a medicinal mushroom and has been used for centuries in China and India to promote vigor, endurance, and longevity. Scientific studies have found that Cordyceps mycelia boost exercise performance. In a double-blind, placebo-controlled trial in adults aged 50 to 75 years, 12 weeks of supplementation with an extract of Cordyceps fermented mycelium delayed fatigue, and it resulted in improved aerobic performance on an exercise test. Another animal study found Cordyceps sinensis mycelia might mimic some of the metabolic benefits of exercise. Supplementation with cordyceps in rats increased exercise endurance despite a lack of training. Significant AMPK activation was thought to be partly responsible for this effect. Potential mechanisms for the exercise-enhancing effects of cordyceps include improved blood sugar regulation, increased insulin sensitivity, and a greater production of ATP, the cell's energy source. Panax ginseng is a popular herbal medicine, and it's used worldwide to increase physical strength and to reduce fatigue. Potential mechanisms for the performance-enhancing effects of ginseng root include improved fat utilization for energy while sparing glycogen, increased levels of the vasodilating molecule nitric oxide, and mild central nervous system stimulation. Multiple clinical trials and animal studies have shown that ginseng improves exercise performance and it prevents fatigue. It may have stronger effects in older and recreational athletes. Ginseng appears to delay exercise-induced fatigue. In a controlled study in healthy male subjects, 8 weeks of supplemental Panax ginseng root prior to exercise on a treadmill decreased the formation of melaldehyde, a marker of oxidative stress. Exercise time to exhaustion was significantly prolonged. 2 types of compound in ginseng, polysaccharides and ginsenosides, are thought to contribute to its fatigue-fighting properties. Ginsenosides are converted to bioactive compounds such as compound K by intestinal bacteria. Compound K possesses anti-cancer, anti-inflammatory, and anti-allergic properties, and it contributes to the health-enhancing effects of ginseng. Fermented ginseng contains compound K, making fermentation one method of enhancing its bioavailability. Found in the mountain areas of Europe, Asia, and North America, Rhodiola rosea is a herb with a long history of use in traditional medicine as an anti-fatigue, anti-stress, and mood-enhancing agent. Studies have also shown rhodiola has positive effects on exercise performance and endurance in humans and animals. Rhodiola is an adaptogen, increasing the body's ability to adapt to physical stress. Rhodiola also increases the utilization of fat for energy. It improves mitochondrial function and it suppresses free radicals. In one control trial in active young men, rhodiola improved endurance exercise performance by reducing perceived effort. Subjects given a single oral dose, 3 mg per kg body weight, or about 200 mg for 150-pound person, of rhodiola, completed a 6-mile time trial on a stationary bicycle, significantly faster than the supplements who were given a placebo. Rhodiola also lowered the heart rate response to submaximal exercise in the study. Another placebo-controlled trial measured the effects of rhodiola extract standardized to contain 3% rosavins and 1% salidroside in 24 participants. The researchers noted endurance exercise capacity improved 1 hour after an acute dose of 200 mg of rhodiola extract. Rhodiola may lessen exercise-induced muscle damage. In a study in male athletes, 4 weeks of rhodiola supplementation prior to exhaustive endurance exercise significantly decreased markers of muscle damage, notably serum levels of creatine kinase, which rise after vigorous exercise, substantially decreased after the rhodiola consumption. For more information on herbs, supplements, and natural treatment plans, check out my website.